Our first module will focus on skeletal muscle, how it contracts, and the fuels that it utilises during exercise. We're not going to have an extensive coverage of how skeletal muscle functions. If you're uncertain about the basics of skeletal muscle physiology, I suggest you go back and review that in any standard textbook of human physiology. But skeletal muscle is characterised by this specialised structure, and at various levels, microscopic levels, you can see the well-organised structure of skeletal muscle. Ultimately, the actin and the myosin filaments within skeletal muscle interact to generate force, and that's under the control of the nervous system. You should review the basic steps and sequence of events involved in muscle contraction. It starts in the motor cortex with activation of the motor centres that control various muscles of the body. There's descending motor activation through the various motor pathways, and finally excitation of the alpha motor neurons that go to specific muscles. The transmission of the electrical impulse across the neuromuscular junction is mediated by the transmitter acetylcholine, and ultimately the excitation of the motor nerve is transferred to excitation of the muscle membrane and the extension of the muscle membrane into the muscle, the T-tubule. The fundamental event in muscle contraction we refer to as excitation-contraction coupling, whereby depolarization of the T-tubule and activation of the calcium release channels from the sarcoplasmic reticulum results in calcium release into the sarcoplasm. Calcium then binds with troponin C, allows the interaction of actin and myosin, and we get the generation of force. ATP, or adenosine triphosphate, the high energy molecule that's utilised at various steps in muscle contraction, is very important, and we'll spend some time looking at the energetics of muscle and the fuels for exercise in our next module. This figure summarises those processes in graphical form, and you can see the various steps involved beyond the neuromuscular junction in the skeletal muscle. We'll come back to this when we look at muscle fatigue in our fifth module, and you can refer back to this figure to see where parts of this process may be affected during fatigue. So again, just to reiterate some of the key steps, we have excitation of the muscle membrane and the T-tubule, release of calcium from the sarcoplasmic reticulum, and during recovery, the resequestration of calcium back into the sarcoplasmic reticulum, the binding of calcium to the thin filament complex, and the interaction of actin and myosin and the hydrolysis of ATP. There are a number of processes within muscle contraction that are dependent upon ATP. Notably, the myosin ATPase is important in the cross-bridge cycle. But other important sources or other important sites of ATP utilization include the sodium-potassium ATPase, which is very important in maintaining the excitability of the sarcolemma, particularly in returning potassium to the muscle after each action potential. Another important ATPase is the calcium ATPase on the sarcoplasmic reticulum. And during relaxation, when calcium is pumped back into the sarcoplasmic reticulum, this enzyme is very active. Studies in rodents where selective inhibitors of the myosin ATPase have been used to reduce the force output of muscle, but during activation there's still activity of the sodium-potassium ATPase and the calcium ATPase, suggest that the ATP utilization due to the myosin ATPase or the force production is about 50 to 60% of the total ATP turnover. But all of these processes are critically dependent upon ATP. If we turn our attention to how ATP is utilized during exercise, we need to just reflect on how in exercise science we express exercise intensity. And that's normally done relative to the maximal oxygen uptake. We'll come back to that concept in our third module on oxygen uptake. But for the moment, if you think of VO2 max as a signpost or a marker of exercise intensity, anything up to 100% of VO2 max is largely aerobic with a small contribution from the so-called anaerobic energy stores. If you exercise at an intensity higher than VO2 max, then clearly the other energy systems, the non-aerobic energy systems, are contributing to that power generation. The muscle ATP concentration is in the order of 5 to 6 millimole per kilo of wet muscle. If you're exercising at 75% of your VO2 max, reasonably moderate intense exercise, if you relied solely on the ATP within your muscle, you'd only be able to go for about 15 seconds. If you increase the exercise intensity to a higher level, say 140% of VO2 max, you'd go for about 6 seconds. And then if you went as hard as you possibly could for a very short period, relying only on the ATP in your muscle, you'd last less than 2 seconds. So clearly, other metabolic pathways in skeletal muscle are activated to generate ATP to maintain muscle contraction. These energy systems traditionally have been divided into two types. The first refers to substrate level phosphorylation, where the ATP is regenerated via the interactions and the exchange of phosphate during a series of reactions directly at the substrate level. The first of these is using creatine phosphate, which is another high-energy phosphate compound stored in skeletal muscle. The other pathway is the so-called anaerobic glycolysis, where glucose units, either from blood glucose or muscle glycogen, are broken down in a series of steps to pyruvate, which can then either be oxidized or converted to lactate. And at high rates of energy expenditure, the conversion of pyruvate to lactate is the dominant pathway. The other metabolic pathway is oxidative phosphorylation. And in this pathway, the metabolism of carbohydrate and fat generate electron donors, which then interact with molecular oxygen in the electron transport chain to establish an electromotive gradient which drives the resynthesis of ATP. This figure summarizes those energy systems. The important processes I mentioned earlier, where ATP is utilized in muscle contraction, and then the other pathways that regenerate ATP from ADP during exercise of varying intensity and various duration. In our next module on fuels on exercise, we'll focus on these various energy systems in more detail. For the moment, however, creatine phosphate and the degradation of carbohydrate to lactate are the so-called anaerobic pathways. And then in terms of oxidative metabolism, the utilization of carbohydrate, lipid, and to a very small extent, protein. It's worth just reflecting on an overview of skeletal muscle energy metabolism. And we'll come back to this, as I said, in the second module on fuels. Here is the utilization of ATP in the cross-bridge cycle. Creatine phosphate serves as a buffer for ATP levels during very intense exercise. The other metabolic pathways involve carbohydrate, where it's degraded through glycolysis and then either converted to lactate or into the mitochondria, where the Krebs cycle and the electron transport chain can generate ATP. The other important fuel during exercise is fat, and this fat can either come from the adipose tissue, triglyceride stores, and the plasma fatty acids, or the intramuscular triglycerides. These are broken down. The fatty acyl-CoAs are transferred into the mitochondria across the mitochondrial membrane. There's a process known as beta oxidation, where these fatty acids can be degraded, enter the Krebs cycle, and in much the same way for carbohydrate, generate ATP through the electron transport chain. As I said, we'll come back to this topic in much more detail in our second module on fuels for exercise. In any standard textbook of human physiology. But skeletal muscle is characterized by this specialized structure, and at various levels, microscopic levels, you can see the well-organized structure of skeletal muscle. Ultimately, the actin and the myosin filaments that control various muscles of the body. There's descending motor activation through the various motor pathways, and finally, excitation of the alpha motor neurons that go to specific muscles. The transmission of the electrical impulse across the neuromuscular junction is mediated by the transmitter acetylcholine, and ultimately the excitation of the motor nerve is transferred to excitation of the muscle membrane and the extension of the muscle membrane into the muscle, the T-tubule. The fundamental event in muscle contraction we refer to as excitation-contraction coupling, whereby discs within skeletal muscle interact to generate force, and that's under the control of the nervous system. You should review the basic steps and sequence of events involved in muscle contraction. It starts in the motor cortex with activation of the motor center. Our first module will focus on skeletal muscle, how it contracts, and the fuels that it utilizes during exercise. We're not going to have an extensive coverage of how skeletal muscle functions. If you're uncertain about the basics of skeletal muscle physiology, I suggest you go back and review that